Hey, Dr. O here, and welcome to Unit 6, the unit on the lymphatic and the immune system. Now, the lymphatic system is kind of a funny system in that it has a structural basis and then it also has the immune component. And so we always like to start the introduction into this unit by talking about the physical makeup of the system. So the lymphatic system is largely involved with returning fluids back to the blood. Now, where are these fluids coming from? Well, they're coming from the capillary beds. So fluid comes into the capillary beds and it leaks out of the capillaries, and then most of it gets taken back up on the venous side of the capillary bed, but not all of it, because there's no system in the physical universe that is 100%. And we have to have, therefore, a mechanism by which we soak up that extra fluid because we don't want it accumulating out there in the tissues. So when we talk about the lymphatic system, we're talking about a network of vessels, the lymphatics, also known as the lymphatic vessels. I also call it the lymphatic vasculature. That language is appropriate as well. And then we have the fluid that's found there, which is called lymph. And then we have these little filtration sites known as the lymph nodes. Now, this is not all that makes up the lymphatic system. We also have some organs and tissues that are going to be largely involved in the immune aspect of the lymphatic system. And these organs are going to house our phagocytic cells and our B cells and T cells that are going to be responsible for launching different levels of immune response. Now, the lymphoid organs and tissues are going to be the spleen, the thymus, the tonsils, which are found in our throat, the lymph nodes, and other lymphoid tissues that we're going to be talking about as we get into the lecture. So let's start off by talking about the spleen. The spleen is a blood-rich organ, and the reason why it's blood-rich is because it filters blood, so we're going to find a lot of blood there at any given moment. It's about the size of the fist. It's located on the left side of the abdominal cavity, kind of just below and behind the stomach, and it is the largest of the lymphoid organs that we have. It does have its own dedicated artery and vein, known as the splenic artery and vein. And the job of the spleen is that it is the site of lymphocyte proliferation. So our B cells and our T cells can actually go there and replicate. So we see, that happening there. And it's also the site of immune surveillance and response. Now, what do we mean by that? Immune surveillance means that these B cells and these T cells that are out in the spleen are looking at what's coming in by way of the blood. And if they see anything that they need to act on, they respond. The other thing that the spleen does for us, and we learned about this in the unit about blood, is that it cleanses the blood of those old red blood cells. It'll also take out any platelets that need to be removed. And the spleen is going to be loaded up with not only our B cells and some T cells that are going to be providing some immune function, but they're loaded up with a bunch of macrophages to remove debris and actually to help with the dispatch of those red blood cells that have basically expired. Now, the spleen does a few more things. It breaks down the red blood cells once we remove them from circulation. And this, again, was a topic that we covered when we talked about red blood cells. And we know that red blood cells get broken down into the heme group and the globin group, and that the globins get broken down to the amino acids, and then the heme gets broken down into iron and bilirubin, and then, the rest of that is, stuff that we've already studied. The other thing that the spleen does is that it will store platelets and monocytes for a time when we need them. Now, we love platelets and monocytes, but they're a little bit like a bad boy or rogue teenagers. You love them, but you don't want to let them run around unattended and without some sort of specific function or a to-do list, because otherwise they can wreak a little bit of havoc in the body. Platelets can accidentally be activated and cause clots if we have too many of them floating around at any given time. And monocytes can also be triggered into action and become macrophages, because monocytes are inactivated macrophages. So we don't really want these cells just, hanging around in large numbers in the body. But when we need them, we want to have them there immediately. So the spleen is where they go to hang out until these things are needed. So if we should injure ourselves and we have a big bleed, not only will local platelets in the area come to the rescue, but the spleen will also get the message that there's been a problem, and it'll release a whole lot of platelets that it's been housing, waiting for that moment. And when we're a fetus and even into the first couple of weeks of life, not much but beyond that, the spleen can also have the capacity to make red blood cells. Now, this does not persist with life, and it's not something that the spleen will do much beyond the first couple of weeks of life, but there is some evidence that the spleen can produce red blood cells when we are a fetus and, like I said, shortly after birth. Now, the spleen is going to be encased in a fibrous capsule, and whenever we see that in my slides or anywhere in your reading, the idea is that whatever's happening inside that organ is nasty. And so we got this big old fibrous capsule to keep the contents in there and protect its neighbors from whatever's going on. And that's certainly true with the spleen, because we've got a large immune cell population in the spleen. And so we want there to be some separation between what's happening inside the spleen and its neighbors, like the stomach, which is clearly focused on digestion and a few other things. Now, when we cut the spleen open and take a peek at what's inside, we're going to notice that there are two distinct regions. One region is called the white pulp and the other one is called the red pulp. So let's take a look at what we're going to find in these distinct regions. Now, the white pulp is where we're going to find a lot of white blood cells, and this is where we see immune function happening. So we have these white blood cells hanging out on reticular fibers, and you might remember reticular fibers from AMP1. It's the reticular fibers are part of the loose connective tissue proper category, and it's called loose because there appears to be open spaces in there. And we're going to see reticular fibers associated with a lot of white blood cells that go into tissue and hang out. And the reason why is because those cells need some place to hang out on. And so the fibers end up being attachment sites for our B cells and our T cells to hang out on in these tissues. So we're going to see the spleen made up of reticular fibers, and it ends up acting like a scaffolding for our white blood cells to basically sit and do their business. I just realized what that sounded like, but you know what? I'm going to leave it. So not that kind of business, the white blood cell kind of business. White pulp is going to be seen clustered around arteries that are coming in, and that kind of makes sense if we think about it, because if the arteries are bringing blood in, then we need to filter that blood. So that's what we see happening with the white pulp. Now, red pulp is the area where we have the old red blood cells as well as pathogens being removed from the blood. And it's going to be rich, this area is rich in red blood cells. And it's also going to be rich in the macrophages that are tearing the red blood cells down and doing the recycle business. It says here the splenic cords, which is basically strands of reticular tissue that are running in between the areas of white pulp. What you're going to find here are these little venous vessels that are rather large, and we want them large because we want to be able to have them take up byproducts of red blood cell metabolism, as well as maybe even some other cells that need to move around the body, like maybe some T cells that are going to start to do a migration. So the way that I think about white pulp is that white pulp is where our white cells are, the white lymphocytes, B cells, and some T cells, and red pulp is where the red cells are undergoing their business. That's a simple way to think of that. I didn't say this earlier, but the spleen is a primary um, lymphoid organ, and the thymus is the other one. And then all the other ones, all the other organs that we're going to talk about, the lymph nodes and tonsils, those are secondary lymphoid organs. So the spleen and the thymus are your primaries. Now let's go ahead and talk about the thymus. Now the thymus gland is found in the inferior part of the neck, the superior part of the thoracic region, just behind the sternum. And its job is to basically educate T cells. That's kind of the short answer with that. So when T cells are first created, they don't really know which way is up. And so they get shipped out to the thymus gland, and the thymus gland basically gives them their education so they understand how the body's supposed to work. And we're going to talk a lot about what T cells are actually being educated on later on in this lecture. Now, the thymus is active early on in our life when we're actually building our immunity up. So when we're first born, we don't have a real robust immune response. We rely upon mother's milk. Our immune system gets engaged and starts to ramp up, and the more cooties that we're exposed to, the stronger our immune system gets. Now, by the time we're an adult, we don't need to have a really active immune response because we basically have encountered a lot of stuff, and our body's largely capable of handling that. So as we age, our immune response tends to decline. We still have a little bit of activity in that thymus gland, but most of the tissue is no longer biologically active, and the capacity of the thymus to educate T cells and do its business slows down a little bit, as do most processes in the body as we age. So thymus gland more or less ages out and turns to fibrous, chunky, non-active stuff tucked behind the sternum. Now, when the thymus is biologically active, what we're going to see in there is that there's going to be lobes. And the lobes have a distinct outer region and a distinct inner region. And there's different things happening there with regard to what the T cells are experiencing. Because remember, the T cells get shipped there in order to go to school. The outside of a lobe, the cortex, is where we're going to find rapidly dividing lymphocytes. So the spleen is someplace where the lymphocytes can go to divide. The thymus is another one where they can go to divide. And so the only place that T cells are made is not necessarily just in bone marrow, but these white blood cells can divide wherever they are. So there's a number of sites where we can create our T cells and our B cells, and the thymus is certainly one of these. Spleen is also another one. All right, so in the cortex, we have our T cells showing up, and they're dividing, and we could call them thymic cells, which is what we see on this slide. The thymic cells are just the T cells that are dividing out there in the cortex, all right? We're also going to find a few macrophages there, and we're going to talk about why we would find some macrophages in the cortex in a little bit. Now, the medulla, we're going to see fewer lymphocytes here, but they're going to be clustered into these regions known as the thymic corpuscles. So what's so special about these guys? Well, what we know about them is that these corpuscles are areas where the regulatory T cells, which is a specific type of T cell, develops. And I don't have it included in my notes, but we can pretty much guess then that there is an additional educational process that is happening in here. In the medulla. There's also educational stuff that's happening in the cortex, but the thing that we absolutely know is happening in the cortex of the thymus lobule is that the T cells are replicating. So that's a little bit about that. Now, when we talk about the immune organs and tissues, we're going to be talking about, oh, it fights the pathogen, it kills the pathogen, it eats the pathogen, it presents the antigen, and all these different things. It's all about, taking out something that's managed to make its way into the body. But when we talk about the thymus gland, we're not going to say those things. And the reason why is that it's different from all the other lymphoid organs in that it does not contribute to immunity or actively contribute to immunity with a defense mechanism. And the reason why this is the case is because it is the job of the thymus gland to educate T cells. So it's an educator. It's an academic setting. It does not directly fight antigens, which is number 2 in our list of information here, a list of reasons why the thymus gland is different than what we would see elsewhere in the body. So its job is basically just to mature those lymphocytes, give them an education, and that's it. It doesn't launch an attack. It doesn't recognize pathogens itself. It doesn't do that. It's all about focusing on getting those T cells to the point where they can do that business for them. Now, because what is happening in the thymus is so important and at the same time possibly very deadly, this is one of the areas in the lecture where we're going to talk about a blood barrier. There's actually a blood thymus barrier, because when the blood shows up, the blood contains a lot of different things. And if some of those things were to be exposed to a developing T cell, the T cell might say, oh, look, there's something for me to kill, without knowing that maybe that's something that is beneficial to the body. So it's really important during the educational process, the maturation process of a T lymphocyte, that the thymus keep what it exposes to that T cell down to a bare minimum. So we're gonna see a blood thymus barrier, something that we don't see in a lot of tissues in the body, but we will see it here. All right, so it doesn't fight antigens. It has and it has that blood thymus barrier in order to regulate what the T cells are being exposed to. The other thing that makes the thymus different than a lot of other tissues in the body is that we will never see any B cells in the thymus. I like to call the thymus University, and it is an exclusive school, and only T cells are invited, and the B cells have to go somewhere else. Harsh, but it is what it is. So there's no B cells going to be found in the thymus. Now there's some language here on this slide that I want to point out. And number 1 says that the thymus has no follicles because it lacks B cells. So a follicle is an area where B cells will take up residence and then proliferate. And I'm going to point that out when we get into areas of the lecture where it becomes relevant. So wherever you see the word follicle, you have to know that a B cell is occupying that follicle. There may be a smattering of T cells, but T cells tend to be on the move. They don't tend to move into tissues and take up residence like the B cells do. And when the B cells do, they tend to live in these structures that are called follicles. All right. The third thing that is different from the thymus gland than from a lot of other organs in the body, including lymphoid organs, is that it has a scaffolding-like structure in there with all of these immune cells to perch on, but it's different than what we see with the spleen or even the liver that have a reticular tissue making up their scaffolding. The soft scaffolding of the thymus gland is actually made up of epithelial cells and not reticular fibers, and that's another way the thymus gland is different. Now, I don't know that that is part of the lecture for my Galen students, but I do like to leave that in there so it just helps paint a clearer picture of what's happening in the thymus gland. So let's move on and talk about lymph nodes. Now, lymph nodes are classified as a secondary lymphoid structure, and doesn't mean they're any less important, because they're very important. So they are the principal secondary lymphoid organs of the body, and there are hundreds of them found throughout the body. In the module for my Galen students, this is they point out that you have them in the neck region, that you have them in the armpit, and you have them in the groin. Those are not the only places that you're going to find them. Literally, lymph nodes are found everywhere. It's just that in those areas, they're a little bit more at the surface and easier to discover. So just pointing that bit out. So they're embedded deep in the connective tissues in clusters along lymphatic vessels. And in fact, a lymphatic vessel will kind of travel along, and then there'll be a lymph node embedded in it. And then a filtration business will take place, and then the lymph will leave by way of a lymphatic vessel, and then it'll continue on until it hits another lymph node. And so lymph can be filtered by a number of lymph nodes before it actually gets returned, before that lymph gets returned into the blood, which is the ultimate destiny of our lymph once it's been cleansed. Talking about where we're going to easily find lymph nodes, some are located close to body surfaces, such as in the inguinal region, the armpit, or the axillary region, and the cervical region. So that's just a little bit about where we can easily find them, but understanding that they are found everywhere in the body. Now we know that one of the functions of the lymph nodes is to filter and cleanse the lymph. And how does it do that? Well, we got a bunch of white blood cells living in there. That's how we do it. Macrophages are going to be hanging out in there. They're going to be destroying microorganisms. And that's definitely one of the cleansing things. But the other thing that's happening in a lymph node, and this is a really important thing, is that we have something called immune activation. Now, immune activation means that we've got like B cells and maybe some T cells that have gone to hang out in that lymph node because they know that the nasty stuff is going to show up there, and it's their job to deal with the nasty stuff. So they have to go where the business is. So immune activation is where the lymph nodes are going to introduce the B cells and the T cells to some pathogen or antigen that has made its way inside the body. So it offers a place for the lymphocytes to become activated and to mount an attack. So the lymph nodes are a place where we're going to do surveillance and response, just like what we talked about in the spleen. All right. I like to talk about the lymph node as a creepy bar scene. And if you think about it, you got a bunch of corn-fed Iowa boys, not to throw Iowa boys under the under the bus or anything, but come from heartland of America, maybe not a lot going on in that small town they're from, and they decide to join the Navy because they want to see the world, and then they've been out at sea for a while, and they come into a port town, and in every port town there's a bunch of bars, and in all of those bars there's action to be had, and so those boys will go into a bar and come out slightly different from what they encountered. Depending on what they encountered, they might be quite a bit different. And that's more or less what's happening in a lymph node. And we could think of our B cells and our T cells as our immune system looking for activation. And so they go into that lymph node where they know where all the action is, and go in there to get it. So creepy bar scene. That's how I like to talk about lymph nodes. Now, the way a lymphatic vessel is put together is not too different from how a vein is put together. And we start off really small. So I use the word lymphatic capillary here because the lymph vasculature actually starts in the capillary bed alongside our blood vessels that are capillaries. And this is the tiniest of the lymphatic vessels. And we want them to be weepy, just like we want our blood capillaries to be weepy, except instead of spilling fluids out, it's the job of the lymphatic vessels to let the fluid in. So they're kind of weepy in reverse. And they have these little, the walls of these small vessels have little mini valves in them that will be forced open when the fluid accumulates and creates a large enough pressure in the interstitial space, and that's what allows the fluid to make its way into the lymphatic vessels there at the capillary bed. Now, I've given you a great deal of detail about how these little mini valves are structured. Basically, you have a collagen fiber anchoring part of the valve system in place while another part can swing free, and that helps hold everything in place, and it also allows an open and a closed scenario. So that's what's happening with those capillaries. So little mini valves that allow stuff in. Now there's going to be valves all along the way because there's no pressure in the lymphatic vasculature. So very much like the veins, it's going to rely upon the muscular pump to move fluid along, which is why it's important that you get up and walk around every now and then. It helps with your blood return, but it also helps move fluid out of the tissues and into the lymphatic vessels, and then keeps that fluid moving along the lymphatic vessels. And as the fluid moves along the vessels, it's going to hit little valves, very much like what we see in veins, and then it'll hold it there until the next wave of fluid and muscular contraction happens, and then it just continues that way until we get into the larger of the lymphatic vessels, where there is a little bit of musculature, I think, if I remember correctly. I don't want to say yes, there is, and that will help move it on into the ducts, where it will then be returned to the blood supply. Now, there is a special type of lymphatic capillary known as a lacteal, and the only place we find these is in the wall of the intestines. It's in the intestinal mucosa. And the purpose of these special lymphatic vessels is to absorb digested fat and then get that into the circulatory system. So fat is difficult to get into our very liquidy blood supply, and it gets stored in these structures that are known as chylomicrons, and they're too big to slip into the capillaries once it comes out of the intestinal lumen. So we have to have another mechanism to pick them up, and the lymphatic vessels are there to do that, and that would be the lacteals that do that. Now, the way that the lymphatic vasculature works is that you have small vessels that are going to join and create larger vessels, and then those will join to create larger vessels yet again. And then we just have this continuous merging, very much like what we see with our venous blood supply, where it just merges from smaller vessels to larger vessels until we're at the very largest of our vessels, and then we're done with our business, which, if we're talking about blood supply, is the superior and inferior vena cava delivering blood to the heart. But we're in the lymphatic system, so we need to talk about the last stop in the lymphatic vasculature, and these are called the lymphatic ducts, and there's two of them. You have one on the right and you have one on the left. The one on the right is called the right lymphatic duct, but the one on the left is not called the left, it's called thoracic duct. Don't ask me why, but there it is. Now, what's going to drain ultimately into the right lymphatic duct? Well, it's going to be all of the lymphatic vasculature that drains the head, the right arm, and the right thorax. All of that gets collected and ultimately drains into the right lymphatic duct. Now, the thoracic duct is going to drain the left side of the head, the left arm, and the left thorax, and everything below the waist. So it's not evenly distributed, just so you know. Now, once the lymph has made it into either one of these ducts, it's then going to rejoin general circulation, the blood circulation. And these two ducts are going to join up at the subclavian vein. So the right thoracic duct joins with the right subclavian vein, the thoracic duct joins with the left subclavian vein, and this fluid is going to be returned to the blood at the subclavian vein, and then it'll be dumped into the superior vena cava, and then there it goes into general circulation again. I know that up until this point I haven't lectured on the images. I've just given you an opportunity to pause and look at them. But I do want to point out that in this image you see the green demonstrating the right side of the body. So that's going to be our right lymphatic duct draining here. And then you see in red the rest of the body being drained by the thoracic duct. Now, there's an additional structure that I want to point out here, and that is called the cisterna chyli. And about half the population has this, half the population does not. And there's no clinical significance whether you have it or whether you don't. It's not like it gives you superpowers. If you have it, it's not like you're diminished in any way if you don't. It's just a normal variation that we see in the population. And it's just this enlargement that we see in the system, and it kind of acts like an interim duct. But whether it's there or not, the fluid that's coming from below the waist is going to make its way into that central thoracic vessel and then make its way ultimately to the thoracic duct, where that gets attached to the left the left subclavian vein, and then enters the blood supply. All right, let's talk a little bit more about lymph nodes. We've already touched base a little bit about them as far as the creepy bar scene goes, but let's talk about how they're put together, because that's important to how they function. Now, they can vary in their shape and size, but most are bean shaped, like a pinto bean or a lima bean, and they're not very big. They're typically less than an inch in length and maybe diameter as well. And they're going to be surrounded by a fibrous capsule. There we go, fibrous capsule. Why is anything surrounded by a fibrous capsule? Because it's doing something nasty in there that it doesn't want to expose its neighboring tissues to. And certainly that's true with the lymph node. Now, the capsule fibers are going to extend inward and they're going to create these little compartments. And whenever you have tissue that is creating compartments, you're going to call that tissue trabeculae. We learned that language when we were studying the bones, and we specifically looking at spongy bones, and we learned that we have trabecular bone. Basically, that's just the arrangement of bone in such a fashion that it created these little open compartments. And that's what we see happening here in the lymph node, is that this fibrous capsule extends inward into the inner part of the capsule and creates these little compartments. Now, these little compartments are going to exist mostly on the outside of our capsule, the cortex, and then there's going to be a distinct inner part which we're going to call the medulla. So let's take a look at what we're going to find in the cortex and what we're going to find in the medulla. Now, the cortex is going to be on the outer part of our lymph node, largely the outer part, and it's going to contain follicles. Now, I told you a word association a moment ago, and I said if I say the word follicles, who's living there? And I hope you remember that it's going to be the B cells. See, B cells are kind of funny. They get released from the bone marrow and they scoot around the body and they're looking for some place to take up residence. And some of them are going to get into a lymph node and they're going to look around and they're going to go, oh, this just looks like home to me. And then they're going to look around in the follicle and they're like, yep, this is it. This is where I'm going to live the rest of my life. And I'm going to have a family here. And this follicle is going to be my little community. And so wherever you see follicle, you know you're going to have B cells living there. All right, so the cortex is going to be loaded up with follicles, with those B cells happily living out their lives, having little barbecues on the weekend, driving the kids around to baseball games, you know, that sort of stuff. Family life, all right? You periodically are going to find a T cell there, but T cells are a little bit like the rambling man. You know, they'll be passing through, they might stay a while, you know, have a burger and, you know, watch the kids play a baseball game, but eventually they're moving on. All right, so deep in the cortex, you're going to find loads of B cells that are dividing, living their best little B cell life. And then you're going to see some T cells that are going to be there kind of doing some of their stuff, but mostly moving on. All right, so that's what we're going to see in the cortex largely. Now, in the medulla, the kind of the central part of our lymph node, we're going to find some B cells. And I'm going to be specific with my language here. A B cell is a cell that has not yet been activated and doesn't know what it's going to kill. It has some idea because it's got a special receptor on it, but perhaps it hasn't been activated yet. T cell could be an activated T cell that's roaming around looking for something to take care of. And then we have a plasma cell, which is an activated B cell and is actively making antibodies. And this is what we're going to see in the medulla. So there's a lot of activity. There's a lot of activity in the follicles, in the cortex, and there's going to be additional activity happening in the medulla as far as an immune response. And it's all predicated upon what is found in the lymph as it filters through the cortex and into the medulla. Now, we have these structures in the lymph node called lymph sinuses, and they're found throughout the lymph node. And these are large lymphatic capillaries that are going to be spanned by the crisscrossing reticular fibers that are creating the scaffolding that helps give the lymph node shape. And the reason why we have these reticular fibers are more robust than collagen fibers. And the reason why we have reticular fibers here is because they act like a soft skeleton and they act like a perch for macrophages and our lymphocytes to sit on and do surveillance. So that's what we have happening here. So when I say creepy bar scene, I mean, imagine if you walked into a bar and you had people that were on all kinds of scaffolding checking you out as you came in. That's pretty creepy, but that's exactly what's happening in a lymph node. So once a B cell gets in there, once a drop of lymph gets in there, the first thing that happens is that the B cell is going to try to find some place to take up residence on this scaffolding so it can do surveillance. And the lymph is going to go by and have everybody check it out. Creepy. Now, I do want to talk about this image because this is an important concept to have with regard to a lymph node. Now, this is a bean-shaped lymph node, and you'll notice that on one side we have 1, 2, 3, 4, 5 lymphatic vessels feeding it. And it's entering through the cortex, and it's making its way into the medulla, where we have 1, and in this case 2, lymph vessels draining it. So we have to ask ourselves, why do we have 5 doors in and only 2 doors out? And the reason is that we want there to be more stuff coming in and lingering once it gets there. And we kind of want to slow it down and not let it escape so easily. Because remember, the whole purpose of getting lymph inside here is to give the B cells, the T cells, and the macrophages an opportunity to see what's in that lymph and then to act on what might be in there. So many doors in, 1 or 2 doors out, and it's all about hanging onto that lymph for a little bit of time to encourage surveillance and response. Now, the tonsils are going to be the simplest of the lymphoid organs, and we're going to see these tonsils proper. They form a ring around the nasopharynx and the oropharynx, pretty much anywhere where we can bring in something into the body. And the reason why we want them there is so that they can do a surveillance and response to any sort of pathogen that we might be inhaling or eating or drinking. All right. Now, unlike the structures that we've talked about up to this point, there is no fibrous capsule in place. And the reason why is that we want this tissue to be exposed directly to the pathogen. So as the pathogen either floats across if we're breathing it in or is washed across as we're swallowing it in our food or drink, we want it to have exposure to the tissue of the tonsils because that is where our immune cells are going to be located. Right there, just right there. No fibrous capsule. Now, we've got a number of tonsils that we're going to talk about, and we've got the palatine tonsils, which are the ones that you see when you open your mouth and you say ah. Those are back there in your throat. You have one further back still located on the tongue, and that's called the lingual tonsil. Then you have some that are called the pharyngeal tonsils, and those are located up high in the nasopharynx, and these are also called the adenoids. And then there's a fourth one that is called the tubal tonsils, and these are located around the opening of the eustachian tube that drains from the middle ear down into the pharynx. Now, for my Galen students, I know that the lab only looks at 3 of these, but I do go ahead and point out the fourth one because it really paints a very clear picture of how we have this tonsil ring around the throat. So let's take a look at a picture. So I'm going to start down here at the bottom. So down here we have the lingual tonsils on our tongue, and then we have the palatine tonsils, and those provide a really good interception point for anything that we eat or drink, which is a big avenue of contamination. And then up here we have the pharyngeal tonsils, aka the adenoids, that will cleanse the air of anything that might be making its way in. And then the tubal tonsils, anything that might be draining from the middle ear or also coming in by way of the nasal passage. So you can see it just forms this interesting ring of lymphoid tissue that helps keep us from allowing anything to make its way either down into deeper structures like the trachea or even into the esophagus and ultimately into the digestive system. Now the tonsils are going to gather and remove pathogens from the food and air that we take in, and they're going to contain follicles. Oop, word alert, who do we find in follicles? A lot of B cells, that's correct. So a lot of B cells are going to be in there, and they're going to be doing their little, barbecues and kids to the baseball game sort of life, and that's what we call germinal centers. And then we're going to find maybe some scattered T cells in there as well. Now, these are not encapsulated, not fully anyway, and which means we want the tissue itself to be exposed to what's going by it. And what we're going to find with these structures is that the surface is not necessarily smooth. There's these undulations or invaginations that dip inside of the lymphoid tissue, and it creates these little cavities or crypts where we can get maybe food particles and certainly bacteria captured in there. Now that is always a very risky proposition, because when you capture bacteria in an area, it could have an opportunity to replicate. And if that bacteria outpaces the body's immune response, then you can end up with a problem. But typically in lymphoid tissue, you have so many lymphatic cells there or lymphocytes there that their presence will outpace anything that the bacteria can actually do to create harm to you. So the payoff is worth it as far as Mother Nature is concerned, to have these tonsillar crypts trap bacteria and have the immune system respond to it. So this creates a great opportunity, very much like what we saw with our lymph nodes, for B cells and T cells to experience a pathogen and become activated. So tonsils are a place where we're going to do surveillance and response. Now, once we get into mucosal passageways, like what we would find in the respiratory tract, the genitourinary tract, or even the digestive tract, we're going to find that we have lymphoid tissue areas there too. And I like to call MALT basically the tonsils that we find in our mucosal tissue, because that's more or less what they are. They're like little tiny tonsils embedded in mucus tissue. All right. Now, when we get into the gut, actually, a large part of our lymphatic tissue is found in the gut. And the reason for that is because that's a big avenue of getting cooties in your body. I mean, we don't walk around all cut up all the time, so that the skin's not a good way to get in, but we shovel all kinds of stuff inside of our mouths. Maybe you bite your nails, maybe you put your pen in your mouth, maybe you're a mouth breather and breathing in. So there's all kinds of things that can make its way into the intestinal tract. So there's a couple of mucosa-associated lymphoid tissue that we're going to talk about, and one of them is the Peyer's patches, and the other one is the appendix. Peyer's patches are really very much like tonsils in the intestines, and that's how I like to say that. So there are these clusters of lymphoid follicles, so we got a lot of B cells in there, that we're going to find located largely in the distal part of the small intestines. And so they could be little nodules, they could be little clusters, kind of like what we see with our tonsils. And I even put in here that they are structurally very similar to tonsils. Now, what are Peyer's patches going to be doing? Pretty much the same thing a tonsil's doing. They're going to destroy bacteria to prevent it from moving along. And because we have B cells in here, and I know we haven't talked about this yet, but B cells not only make antibodies, but some B cells become memory cells. We're actually contributing to our adaptive immune response. So wherever you have B cells, I'll go ahead and say this now, wherever you have B cells, wherever you have follicles that contain B cells, that's where B cells live, you're always going to have an opportunity to create memory cells and enhance the adaptive immune response with that memory. And Peyer's patches is one of them, along with our tonsils and our lymph nodes and any place else that we're going to have B cells live in. Now, personally, I think the appendix gets a really bad rap because I think it's undervalued in what it does. But let's go ahead and talk about it. I personally am a big fan of the appendix. I got mine. I'd like to hang on to it for the rest of my life. The appendix is considered part of MALT because it is found on the first part of the large intestines, just as the intestinal tract stops being small intestine and becomes large intestine. It's attached to the part of the large intestine that's known as the cecum. And what we're going to find here is a large number of lymphoid follicles, which we know means B cells, which we now know means we're going to contribute to our memory. the adaptive immune system. So we have the ability to make antibodies and deal with an infection and remember that infection later. So that's all good news. Now, the other thing that it does, in addition to destroying bacteria and preventing them from getting into the body and making memory cells, is, I know it's not on this slide, but we have since discovered that because it's like a little sac organ, there's bacteria that live there that can be utilized to reinoculate the gut if there should be a disturbance in the microbiome of the gut. So the more we study the appendix, the more we learn about the amazing things that it's doing for us. Now, you can live without the appendix. Plenty of people do, particularly when it gets bad. But, you know, it's always better to have it because if Mother Nature put it there, there's probably a reason for that. Now, bone marrow is going to be another one of our lymphoid tissues. Yellow marrow, not so much because it's largely fat and it tends to get used for energy storage. Although, interestingly enough, if the body needs it to, it can be converted back into red marrow, which, you know, amazing body, hello. But, you know, as long as it's in the yellow marrow, it's going to be basically fat and get utilized for energy when we need it. If we should starve, if for some reason we're starving. Now, the red marrow is our biologically active marrow when it comes to making the formed elements of the blood. And this is going to be where our B cells originate and where they mature. It's going to also be the site of where the T cells originate, but the T cells get shipped off to Thymus University. So once the T cell is created, it just, you know, gets shipped off and has to go somewhere else. But bone marrow, red marrow particularly, is considered part of the lymphatic system because of its involvement in making the lymphocytes. Now, in this section, we're going to talk about some of the amazing ways that the immune system can launch its attack. So when we think about the immune system, we're going to have what we call the innate or non-specific defenses, and then we're going to have what we would call the adaptive or the specific defenses. And I know that I've used that language adaptive already when talking about B cells and their ability to make memory cells, and T cells and their ability to make memory cells. But we're going to talk a lot more about all of this moving forward. So our innate defenses are going to be basically our first line defense and our second line of defense. And then the adaptive is going to be what we would call the third line of defense. Now, our first line defense is pretty simple. It's basically the skin and the mucosal membranes. These act like a wall, and they are effective at keeping things out and protecting what's on the inside. And let's say that they weren't. Let's say that we had a breach in the barrier. So maybe you got a little cut, maybe got a little nick in your mucosal membrane because you ate a tostada chip, something like that. And something was able to get inside the body. Then what? Well, then you have what's called the second line of defense. And this is where we see entering into the equation things like antimicrobial proteins. We'll talk about those. Phagocytes. We'll talk about who makes up the phagocytes in the innate defenses, and other sorts of cells that are going to do things like create inflammation. So we've got that happening. And then if the bacteria or the pathogen is capable of getting past both the first and the second line defenses successfully and find some place to start to take up residence, well, then we have the third line defense. And this is where the body detects that there's something that's there that shouldn't be, learns about it, and then launches an attack, and then remembers that attack. So let's get into all of this. Now, as I said a moment ago, the first line of defense is basically going to be the barriers, the walls that the body creates. And that's going to involve the skin and the mucosa membranes, as well as everything that they make, because the skin and the mucosal membranes make things that are very hostile to many pathogens. So by virtue of the fact that we have a barrier in place, that acts to repel most microorganisms. In the skin and in parts of our mucous membranes, we actually have keratin, which is a protein that is resistant to weak acids and weak bases, which might be being secreted by bacteria in order to break down our defenses. It's also resistant to any sort of enzymes or toxins that a bacteria might be releasing to further break down our defenses. So keratin's pretty tough stuff. The mucosal membranes also provide similar mechanical barriers. So some of our mucosa do have keratin. We do have keratinized mucosa in our mouth, for example. And then we also have different secretions that can, for example, change acidity and make it hostile to certain microorganisms. So there's a lot to first-line barriers. Besides just being a wall, there's chemical warfare going on here too. So what sort of chemicals are we talking about when we talk about the first line barriers? Well, I mentioned acidity a moment ago. And when we were learning about pH of body tissues in A&P1, you learned that skin comes in at a very acidic pH of 5. And in fact, we call the skin's natural acidity the acid mantle. And the whole purpose of that is that interestingly enough, most microorganisms can't survive at a pH of 5. They can't survive at a pH much less beyond 5.25. So Mother Nature has kind of given us the advantage with our slightly acidic skin. So kudos to Mother Nature on that one. In addition, certain parts of our first-line defenses can make enzymes that are hostile to pathogens as well. And we would call that, well, we just call them enzymes. So for example, in the saliva contains an enzyme known as lysozyme that is hostile to bacteria and can break it down. In the respiratory mucosa, we also have lysozyme, and we also see this in our tears, our lacrimal fluids. So these are all first-line structures that are going to interface with the outside world before whatever they're doing are going to allow something inside the body. Inside the stomach, which is technically also a first-line barrier, because remember the intestinal lining is a structure interfacing with the outside world, even though we think of it as inside there, it is outside. The stomach lining has a bunch of enzymes as well as acid that can kill many microorganisms. So there's a lot going on with first line defenses as far as barriers and chemical warfare. We also have mucin, which is a sticky substance that is lining the digestive tract and the respiratory tract. It can actually trap microorganisms, and then they can be swept out of those passageways or swallowed and then make its way to more hostile environments. We have antimicrobial proteins called defensins that are secreted and can do damage to certain microbes. And for example, lipids in sebum can be prohibitive to certain microbes, as is dermacidin, which is a type of antimicrobial protein found in sweat. Now, the respiratory tract has some interesting modifications that help stop pathogens, and one of the things that it has are those ciliated pseudostratified columnar epithelial cells. And in between those are a bunch of goblet cells producing mucus. So what we have here are these columnar cells, which have cilia on them, and then we have mucus cells that are goblet cells that are producing a bunch of mucus. That mucus gets swept around by the cilia, and so the mucus ends up acting like a trap that will trap dust or bacteria or whatever is being breathed in, and then the cilia will help sweep that mucus along with whatever's trapped in it out to the throat or maybe into the mouth, and we have an opportunity to either cough it out, spit it out, or swallow it, where whatever might be trapped in that mucus is going to be exposed to the acid and enzymes in the stomach, and then most likely be taken care of. All right, so surface barriers are pretty effective, but when we get a nick or a cut or some sort of a breach in the barrier, then we have to rely upon something other than first line defenses to protect the body, and that's where we get into what we call second line defenses. Second line defenses are going to be these defenses that are going to protect the tissues inside the body. So let's have a look at those. So we've talked about our first line defenses. We know it's the barriers, which are going to be the skin and the mucosal membranes, plus whatever sorts of chemicals that they make, and there's a number of those. But what if our pathogen gets past that? What's the next thing they're going to encounter? Well, that's going to be the second line defenses, and these are going to be cells and chemicals that are found inside the body. So these will be things that we find in the tissues or floating around in the blood. Now, this is part of the innate system as well. So basically it means it's nonspecific. And it's going to involve things like phagocytes, and there's a number of those that fall in this category, a number of cells that are classified as phagocytes. We have a special type of a white cell called a natural killer cell. We'll learn a little bit about what that does. And then we have the inflammatory response. And this is going to involve a number of cells and the chemicals that they release once they're activated. So the sorts of cells that are going to be releasing chemicals that are going to fan the flames of the inflammatory response will be macrophages, mast cells, which are a type of a white blood cell loaded with histamine that take up residence in the tissues. Other types of white blood cells like neutrophils, basophils, eosinophils, all those guys we learned about in the blood unit, and all the chemicals that they release. And there's different classifications of chemicals that we're just generically for now going to call inflammatory chemicals. There's also some antimicrobial proteins, and there's a couple of classes of proteins that we want you to know about, and those are interferons and complement. And then we have good old fever. So let's get into talking about each of these. Now, as I just mentioned, phagocytes are going to be one of the second line defenses that the body has to fight pathogens. And a phagocyte is basically some sort of a white blood cell that has the ability to go up to a pathogen, recognize the antigen is problematic, and eat it. Eats that pathogen and destroys it in the process. And our buddy, the neutrophil, is one such of a phagocyte. You might remember the neutrophil from our study of the blood, and this is the most abundant phagocyte that we have. So if we were to look at a blood smear, we'd see tons of red blood cells, we'd see a few platelets, and if we're going to see a white blood cell, chances are we're going to see one or two of these neutrophils floating around. Now, when you have the initial onset of an infection, neutrophils are going to be the EMTs. They are the first responders when there's an infection. So they're going to show up, and they're going to show up fast, and they're going to show up in abundance. Now, we love them for doing that, but the problem with neutrophils is that they die in the fight. So they'll go up to the pathogen, they'll consume it, and then somewhere in there they're going to die. So they don't provide any long-term resistance to pathogens. They're just let's get in there and get the job done, kill them, and then let the body pick up the rest of the pieces, sort of a white blood cell. But they are our first responders, so we have to appreciate what they're doing for us. Macrophages are another type of phagocyte that's going to be doing that cell eating and destruction. And you might remember that macrophages were the cells formerly known as monocytes. So once a monocyte gets triggered, either by inflammatory chemicals or by the presence of an antigen, it's going to turn into this voracious eater known as a macrophage. Now, macrophages have a little bit of variability as to their behavior. So we have free macrophages that will wander around and through tissues, or we have some that will take up permanent residence in some organs and live out their entire lives there. So we know that we have monocytes that are freely moving around in the blood as blood makes its circulation, and then it can follow the chemical trail set forth from injured tissue and then get stimulated and become macrophages. That would be an example of a free macrophage. Another example of a free macrophage are the alveolar macrophages that are free to wander around all throughout the lung tissue, dealing with whatever might have made it all the way into those small air sacs. So those are examples of free macrophages. Now, we've studied at least one fixed macrophage, maybe even two, and these are macrophages that basically decide that they're going to live the entirety of their lives in a particular area of an organ, usually not traveling very far from its locale. And an example of that is going to be the microglia cell in the brain. So you might remember from AMP1 that we studied those special support cells that live in the brain and the spinal cord, the central nervous system, and one of those was the microglial cell, or the microglia. And those are a macrophage that has decided to live in the brain and spinal cord, and its job is to wander around and clean up any cellular debris or metabolites that have kind of are free floating around in the tissues, any toxins that might have made its way in, and basically keep that environment really clean. Another example of a fixed macrophage is a particular type of macrophage that lives in the liver, and this is called a stellate macrophage. And the reason why it's called stellate is because it looks like a star. Stellar means star. So these are a couple of examples of how macrophages can be somewhat variable in their performance in the body, but wherever they are, they're going to be the big eaters. So we've talked a lot about how these two cells are the big eaters and they eat pathogens and all that sort of stuff. So how does that actually happen? How does it know what to do and how does it go about doing it? Well, the process of phagocytosis starts with the phagocyte, whether it's a macrophage or a neutrophil or some other phagocyte. Recognizes and adheres to a pathogen's carbohydrate signature, because you remember that all cells, all microbes are going to have chemicals on their surface, are going to have molecules on their surface, they'll have receptors on their surface, and all of these are communicating with the outside world, and sometimes the outside world is capable of reading those and interpreting them to have something meaningful, such as hey, this doesn't belong here. So we would classify some of those as antigens. Typically antigens are proteins, but it is possible that you could have a carbohydrate signature that is basically a name tag that says, hi, my name is, and yes, I belong here, or no, I don't. So it's just another signifier that exists on the surface of certain pathogens or maybe even certain cells. Now, the thing about microorganisms is that some of them have been, they've kind of gotten wise to what our bodies are capable of doing. And so they have the capacity to hide that carbohydrate signature, and that helps them slip past a white blood cell, like a phagocyte. So how does the body deal with that? Well, the phagocyte is looking for something to tell it to go ahead and eat this thing. And if that something is hidden, then the body, in its wisdom, is capable of adding a molecule to that sneaky little microbe so that the phagocyte understands that that is an invader and needs to be taken out. And this is a process known as opsonization. So this is when the immune system is going to use something like maybe an antibiotic, excuse me, not an antibiotic, an antibody, or maybe something like a complement protein, which we really haven't looked at. But these are chemicals that the immune system will attach to this microbe that's trying to hide, and it will act as either something like a handle or a signifier that lets the white blood cell know that this is the thing to eat. So let's take a look at an example. So let's say that this is our microbe, and on the surface we've got these little X markers, and that's not something that needs to be in the body. So here comes our white blood cell, and it's going to look at those little X markers, and it's going to make a determination that, uh-oh, this is not where it's supposed to be, and it will eat it. So that's basically how that works. But sometimes these sneaky little microbes will put a little cloaking apparatus around them and then try to hide. And so this is what we see happening here. This little microbe has tried to sneak past the white blood cell, and the white blood cells is a little bit confused by what it sees. But the immune system recognizes that this is a problem. So what will happen is that it will attach a chemical to it, and here we have kind of a chemical attached to it. In this case, it's an antibody, and this is an act called opsonization. So the immune system has added something to this microbe, so now the white blood cell understands that this is a problem and it needs to be taken out. So in that previous image, what we saw was a white blood cell, and it was sending out its little extender pseudopods to latch on to some sort of a microbe and bring it in, kind of like a tractor beam from the Starship Enterprise. So yeah, that's what we saw. And that is basically the second thing that's going to happen in phagocytosis. You're going to have this white blood cell, this phagocyte, send out an extension. It's going to basically create a little arm or a little leg, or however you want to call that, some sort of extremity. And it's going to send that out, and it will bind that particle, and then it's going to, that pathogen, let's just call it that, and then it's going to bring it in, and when it does, the cell is going to surround it with its plasma membrane, and then that plasma membrane surrounding the pathogen is going to break off and create a little vesicle inside of the cell. And once we have that pathogen in a vesicle inside of our white blood cell, we would call that vesicle a phagosome. Because if you remember from our study of cells, cells don't have stomachs. So they basically have to create one when they want to do something like digest something. All right, so they create this little pseudo-stomach known as a phagosome. Now, because the only thing that's in there is a pathogen, the cell is going to have to bring in some enzymes. So it's going to bring over a lysosome, and that's another vesicle that contains some digestive or caustic enzymes, and that's going to merge with the phagosome, and it's going to create a structure known as the phagolysosome. So that's basically the pathogen and the enzymes that are about to destroy it. Now, the phagolysosome is, like I said, acidified, going to have a bunch of enzymes in it, and this is going to digest that pathogen and break it down into little particles. And the phagocyte is going to kind of sift through the particles, and anything that it can utilize it will, and anything that it can't is going to be eliminated from the inside of the pathogen in an act called exocytosis. So let's look at an animation that goes through all of these steps. So here's our little pathogen, and here comes our white blood cell, and the white blood cell is going to eat this thing. And when it does, it has to put it in a little pseudo-stomach that's called a phagosome, but there's nothing there to digest it. So the white blood cell has to bring in some enzymes by way of a lysosome. So this is our lysosome with the enzymes in there. And then the lysosome and the phagosome are going to merge, and we're going to dump the enzymes in there. It's going to do some digestion. And at this point, we're going to call this combination of pathogen and lysosomes the phagolysosome. Okay. And then after the enzymes have done their business, we end up with little pathogen parts. And if there's anything that can be used by the white blood cell, it'll be extracted, and anything that isn't will be ushered out of the white blood cell in an act known as exocytosis. So we have another white blood cell, but this one is not a phagocyte. This is going to do something a little bit different. So we have the natural killer cells. Like I said, it's a white blood cell, and it has a bunch of chemicals in it that it's going to release. And its target is either going to be a cancer cell, which has a whole bunch of crazy markers on its surface, or a virus-infected cell. And so that brings us to kind of an interesting little side discussion, which I'm going to go ahead and talk about right here. You see in the body, when a virus invades a cell, one of the things that the virus wants to do is get inside of the cell's DNA and deposit the viral DNA in there and kind of disrupt the normal reproductive cycle and mechanisms involved in the tissue cell. And that basically provides a platform where the virus can replicate, but it's probably going to kill the host in the process. But if we end up with 100 more viral cells by killing one host cell, then so what? The virus got to replicate and propagate, and it's in a stronger position now. So when a body cell gets infected by a virus, it knows it doesn't stand a chance. So it actually sends out a chemical signal that says, I've been infected by a virus, somebody needs to come and kill me. And natural killer cells are one of the cells that are going to do that. All right. So the way that a natural killer cell works is that it's going to largely attack cells that lack the self receptor. It doesn't have that self signal on there that identifies it as you, as a cell that's supposed to be in the body. And it does its killing largely by doing what's called programmed cell death. So it's basically going to speed up the date when a cell dies. So it kind of, basically we can think of it as going in and changing its programming, and then instead of it living for, I'm just picking a number, 120 days, it may just live a few more hours and then it will just die. So that's kind of how the natural killer cell works. And if you think about it, natural killer cell sounds very dramatic, but in the effect, what it does is it's kind of quiet, very stealthy, but it is effective, and we're glad that we have them. And then in addition to programmed cell death, apoptosis, it's also going to secrete a bunch of chemicals that are going to enhance the inflammatory response. Now at this point, pretty much everything that's going to happen is going to involve releasing chemicals that are going to enhance the inflammatory response. So with the inflammatory response, there's a whole lot of things that can happen. And but what I would like for us to think of as these chemicals of the inflammatory response, think of them as that that kid that had the party the first time his parents left the house for the weekend. So he's, maybe the 16-year-old that gets left alone over a weekend for the very first time, and he wants to have like 20 friends over and have this really killer party with no adults around. But the next thing he knows, those 20 friends have told another 10 friends each, and then those 10 friends have told another 10 people, and the next thing you know, this kid's got 300 people at his house, and some of whom he knows and some of whom that he doesn't. That's very much like what happens with the inflammatory response. You have the initial players who send out a bunch of invitations to other immune responders in the body, and then they show up and then they send out invitations too, and the next thing you know, you've got this big out of control house party going on that's trying to take care of the pathogen that got into your body. So that's basically the inflammatory response in a nutshell. Now, an important function that the cells of the immune system are going to do is something called surveillance. And that basically means that these immune cells, even whether it's second line or even if it's the adaptive immune system, they're always going to be scanning the body tissues and the blood looking for problematic cells. But our macrophages and certain other cells are going to go, we know, to certain areas of the body where the chances of them encountering a pathogen are really good. And these are particularly nasty areas like the lymph nodes, because we know the lymph nodes are going to be filtering lymph, and lymph tends to be a little bit contaminated because it's out there in the body tissues where we have metabolites of cellular processes and maybe cell debris and cell parts and maybe some pathogens or pathogen parts. So a lot of garbage there in the interstitial fluid that can make its way into the lymph and then consequently the lymph node. But that's good for immune surveillance and response. The lungs are another place, because this is an area where we are starting to interface with the outside world, and we know the outside world is a little bit nasty. So wherever you have an opportunity to engage with the outside world, the body is going to put a strong immune presence there. So the lungs will be a place. The liver is a place. The spleen, and we could even include the digestive tract in this as well. So the cells that are going to be involved in immune surveillance, at least the second line defense cells, are going to be those natural killer cells, our buddies, the macrophages. Dendritic cells, remember those players? We learned about the cell of Langerhans that lives in the stratum spinosum layer of the epidermis. That is a dendritic cell. And to some degree, the cytotoxic T cell. Now, the cytotoxic T cell is not a second-line defense cell, but it is a cell that will roam around and do immune surveillance, just kind of checking the environment and seeing if that everything is as it should be. The cytotoxic T cell is actually part of the adaptive immune response. And while I'm talking about that, what we're going to start to see between the second line and the third line defense is that there are points where we're going to have overlap. And as far as who is doing surveillance, we have a point of overlap here where we have a T cell, adaptive immune cell, doing surveillance. But that's what happens. All right, so let's switch gears a little bit and let's talk about the antimicrobial proteins, interferons, and complement. So antimicrobial proteins are going to enhance the innate defense, and they do this by attacking microorganisms directly or by hindering the microorganism's ability to reproduce. And that's a big thing there, because like I said, viruses like to get inside of their host cell and then replicate. Because if one viral cell can get into a host cell and create an additional 100 or more of itself, then it wins the battle. In that case, might not win the war ultimately, but it will win the battle. So if we can stop it there, then we stand a good chance of winning the war as well as winning the battle. So let's take a look at each one of these interferon and complement proteins and see what's happening with these guys. Now, interferons are a classification of chemicals, and they're going to be made of proteins, and their job is basically to interfere with a virus's ability to reproduce. And it's interesting how it does that. So interferons don't actually do the reproductive interference, but they act more like a warning message to neighboring cells. So how does all this work? Well, when a cell gets infected with a virus, the cell that is infected with the virus releases the interferons. And the interferons aren't going to help the cell that's infected with the virus. It's going to warn the cells around the infected cell that there is a problem in the community. And that gives the healthy cells an opportunity to put up some sort of a protective mechanism to prevent the virus from invading it. So basically, our tissues were largely unsuspecting that anything was wrong. A virus sneaks in, gets inside that DNA, starts doing its little, viral business, and the cell's like, oh, great, here we go. And it's going to release interferons, which are going to be distributed around it, and then the neighboring cells say, oh, geez, there's an interferon. So somebody's been infected, I better put up the shields so that nobody can get in here too. So let's take a look at what's actually happening with this, because, I've got an animation. Let's take a look. So let's say that we have a couple of cells out here in the tissues, and then along comes a virus. There's our virus, and it's going to insert its RNA inside the cell, technically embed it within the nucleus, and then it's going to start to replicate. And what the cell is going to do once that happens is it's going to produce interferon. So this is the infected cell producing the interferon, and the interferon is not for the cell that was infected, but for its neighbor. So the interferon is going to be released from the infected cell, and it's going to merge with this receptor on the neighboring cell, and then the neighboring cell is going to say, uh-oh, there's a virus in the neighborhood, I better put up a shield. And so now it's got its own antiviral protein mechanism in place. Because what the neighboring cell knows is that if there's a virus in the neighborhood, that virus is going to replicate, and then eventually it's going to come knocking at its door. And when it does, the neighboring cell is going to be ready for it with its shield, and it will not allow that virus in. So interferon basically interferes with the normal cycle of a virus. Kind of like the name implies. Now, interferons are actually pretty simple classification of chemicals as far as how they function. Complement is not. So I'm going to give you the dirty story, the detailed dirty story of complement, but I think you just generally need to remember a couple of things about what's happening with complement. So when we talk about the complement system, we're basically talking about roughly 20 blood proteins that are normally circulating in the blood in an inactive form. And we're going to have proteins that are called C1 through 9, and then we have a bunch of factors with some letters after it, and then we have some regulatory proteins that are involved in this. All of these are made in the liver because, you know, liver is up in everybody's business. And what ends up happening is that these complement proteins will be triggered by enzymes or in a I'll say it that way, they get triggered by enzymes to become active, and each step in the cascade of chemical transfiguration, I'm going to use that word, transformation, is going to lead to the next step in the cascade until we get our final end product that is going to have the ability to kill the pathogen. So that's the short story of the complement. We're going to actually look at a little bit more detail in a minute. So complement then, we could say, provides a major mechanism for destroying foreign substances, because up until this point we haven't really talked about destroying anything directly other than, you know, releasing some chemicals and program cell death and that sort of thing. But this actually creates a mechanism, you heard me correctly, that is going to kill the pathogen. All right. And of course, once it's activated, guess what else gets activated? Additional chemicals of inflammation. So we're going to continue to fan the flames of the inflammatory response. Okay, so let's take a look at what's happening here with complement. Now, even though complement itself is a second-line defense, the classical pathway that complement gets activated actually slips us into the world of the third line defense. So that's why I have third line up here in the title of my slide, is because we're going to engage some third line innate defenses with complement. So like I told you, we're going to start to see points of crossover between second line defenses, which are innate defenses, and adaptive defenses, which are specialized defenses in the third line of defense. So this is another one of those examples where we're going to see this. So in the classical pathway of complement stimulation, what ends up happening is we have antibodies, which are an innate defense, are going to bind to a pathogen, and then our complement protein chemical is going to bind to the antibody. That is your classical presentation. So basically what's going to happen here is we have our invader, and then we're going to, you know, have antibodies attached, and then we're going to basically have our complement protein come along, and then we're going to call in all the players of the immune system to come in and do their best and kill this thing. So that's kind of the general idea of the classical pathway. You have an antibody that binds to the pathogen first, and then the complement protein. Now you'll notice on my slide that I've gone back to second line defense. And the reason why is that we're not going to have the involvement of antibodies in this second pathway, this alternative pathway. In this scenario, what ends up happening is we have our little pathogen, and instead of having an antibody, which is a third line defense player, come in and attach to our pathogen first, we're just going to directly apply that complement protein right to the pathogen. And then we're just going to proceed down the same pathway and call the same players in. So we have two ways to start, but we're basically going to finish the same way, regardless of which starting mechanism we use. So whether we have the antibody and the complement protein together, or just a complement protein, the end effect is the same. So there are reasons why we would have one pathway versus the other. So for example, we wouldn't necessarily need an antibody if our microorganism lacked some sort of an inhibitor on its cell surface. So, you know, keep in mind this is all happening at the microscopic level, and there's a great deal of assessing of these microbes that are happening. And if certain things are replaced, well, then we can take this pathway, but if those things are absent, then we can follow this other pathway in order to get to our final result. So that's basically what we're looking at here. So if our microbe doesn't have certain inhibitors that are resistant to our attempts to kill it, then we get to follow the alternative pathway. But if those things are there, then we're going to have to attach the antibody. So once complement has been affixed to a pathogen, it's going to activate a process, a cascade of events, where one protein's creation creates the next complement protein, activates the next complement protein in the sequence. And whether we start with the classic pathway, which involves the antibody and the complement affixing to that, or the alternative pathway where the complement directly attaches to the pathogen, both of those processes are going to converge on a step that involves the complement protein number 3. So it doesn't matter how you start, very quickly, both of those starts are going to take us to the same point in the cascade, and that is where we have the creation of the third complement protein. Now this protein, once it gets created, is going to immediately get cleaved in two, and it's going to be, because it's a big protein molecule, and part of it's going to be called complement 3A, and the other one's going to be called complement 3B. And splitting the third complement protein is, like I said, that common pathway, that common point in either the classical pathway or the alternative pathway. And of course, it's going to, once we get to that point, we're going to have the release of additional inflammatory chemicals, and then that's going to bring in more phagocytes and cause the cells to be destroyed, and, you know, just more of the same, more of the more. Now, complement itself is going to be responsible for destroying the cell. It's going to be responsible for cell lysis. So once we have activated that third complement protein, then the cascade that happens is going to take us down the pathway through a number of steps, and what happens is that each protein leads to the creation of a specialized protein that at the end will be assembled into this ring-like structure known as a membrane attack complex, which we abbreviate as MAC. And this membrane attack complex, like I said, it forms a ring-shaped structure. These are made out of different protein molecules that create this ring-shaped structure. And that ring-shaped structure is going to basically go up and punch a hole in our pathogen and basically hold that hole open. And in a water environment, that is death. Because what will happen is fluid will flood inside of that pathogen and cause the pathogen to basically explode. So I like to call that membrane attack complex a MAC, and that it does the Big Mac attack. That's how I remember that. The MAC attack. Punches a hole in it. Kills it. Now, activation of that third protein is going to cause opsonization, so we could end up attaching additional complement to a pathogen or pathogens in the area, or the attachment of antibodies to other pathogens in the area. And the 3A that gets created as the third complement breaks apart, this is actually going to going to be the part that amplifies the inflammatory response. So the 3A component that gets created once the third protein gets divided in two or cleaved in two is going to stimulate mast cells and basophils to release histamine, which is actually a chemical of inflammation. It's one of those inflammatory stimulating chemicals. And then that's going to attract neutrophils and macrophages to the area, and off we go. So now we've got a bigger response than we had just a second ago, thanks to all those inflammatory chemicals that are being released. Now, this is an image from your textbook, and it goes into a great deal of detail talking about what's happening here. So let me give you a little tour. So this green thing is a pathogen, and it's got the little yellow antigen right here, and these little Y-shaped structures are antibodies that are attached to it. So let me ask you this. If we have antibodies attached to the antigen, and then we have our complement protein attached to that, are we looking at the classical pathway, or are we looking at the alternative pathway? Well, I'll tell you the classical pathway, because that's going to be the one that involves the antibodies. And that is one of those crossover points where we go from second line defense to now involving some component of third line defense, which in this case is going to be that antibody. All right, so here we've got the first complement protein affixed to the antibodies, and then what's going to happen is a cascade of events. So this first complement protein is basically going to so it binds to the antigen, and it's going to cause complement protein C2 and C4 to split in two. So it basically acts like an enzyme. We can almost think of it as acting like an enzyme that's going to cause some other complement proteins that are floating around to be activated. So we're going to split the C2 and the C4, and these two are going to come together and create that C3. Now, C3 was important a moment ago because we talked about whether we start with the classical pathway or the alternative pathway, they both very quickly end up right here. So we're showing you the classical pathway, but if we had started off with the alternative pathway, this point would be identical. So from this point on, it doesn't matter how we started the process, it's going to be the same. So we're going to have C3, and that gets created. The third complement protein gets created, and then that will be acted on by an enzyme that's going to cause it to be cleaved into two separate subunits, which we're going to just randomly call C3A and C3B, so we can kind of keep track of it. Now, this is an important point here because the C3A is going to attract phagocytes to the area. So we talked a little bit about that, sending out those signals that are going to say, hey, we're having a party, come join us. So here comes the phagocytes. And then C3 is going to continue on down, and it's going to bind to another protein. And then off we go until we end up creating some additional proteins through enzymatic reaction. And then that's going to lead us to our MAC, our membrane attack complex. So we're not showing all of these proteins being created in this cascade, but you get the idea that one's going to lead to another and lead to another, and maybe we have some stimulation along the way in some of these intermediate steps, but the ultimate goal is to assemble this ring of proteins that is going to have the ability to punch a hole in the side of our pathogen right up here. So this thing is going to whip back around, it's going to attach to this thing, but we're not going to show that literally happening. We're going to put the pathogen right down here, and the pathogen is basically going to fill with fluid because this membrane attack complex, this circular protein structure, is going to basically punch a hole in the side of our pathogen, hold that hole open while water floods in, and cause our pathogen to spring leaks all over the place and die. So that's complement. So what do you take away from all of this? Well, I would have an understanding of the difference between the classical pathway and the alternative pathway. It either involves application of complement to the antibody or complement directly to the pathogen. So that's the difference there. I would have some idea that this C3, this third complement protein created, is the point at which the two different initiations of the complement pathway will converge, and that everything down the pathway will be the same. And then I would know that the result is this. We're looking for that MAC, that membrane attack protein creation, and then I would know what that does. Pokes holes in the pathogen. So we're getting kind of to the end of all of our second line defenses, but does it mean they're any less important? And this one certainly isn't. Inflammatory process, inflammation. So inflammation gets triggered whenever the body tissues get injured. There's a number of things that happen when body tissues get injured. And basically, the body recognizes that cell parts need to be inside of cells and not on the outside. And just like us, when we see When we see parts of the body on the outside, when we know that they should be on the inside, we all have a tendency to get a little bit wound up. So that's the same thing is true inside the body, and it can actually trigger cells to release inflammatory chemicals in order to get the immune response going and take care of the tissues that are remaining, and then deal with any pathogens that might have invaded. So when tissues get injured, the inflammatory response gets initiated, and it doesn't matter what that injury is. It might be trauma, like a blunt force injury due to a car accident or falling out of a tree, or just taking a fall going down the stairs, something like that. Maybe you sustained a burn, or you have a chemical that was spilled on you, or maybe you do have an invasion of a pathogen. It really doesn't matter what the mechanism of injury is. Once the cells are injured, the inflammatory response gets triggered and off it goes. Now, we do like inflammation because it does have a number of benefits. Number one, it prevents the spread of pathogens and any sort of damaging agents that it might be releasing. Because pathogens do release toxins. They're just not coming in here and hoping to share in our picnic. They want to take over the world. And they do that by disrupting our tissues and doing damage. That's how they work. So one of the things that inflammation does is it helps contain those pathogens in the area, and it's doing that by bringing a lot of cells of immunity, which might be killing the pathogens or consuming the toxins and cell debris. So there's a number of ways that the body's going to do that. Inflammation leads to those things that are basically going to clean up the area and help prevent the spread of damaging agents. And in the process of cleaning up the area, it's going to do disposal. So it's going to get rid of the cellular debris from the damaged cells, and hopefully the damaged pathogens. It'll clean up those parts too, if just not take the pathogen out completely. The other thing that inflammation does is that it alerts the adaptive immune system. So there is crossover between the second line defense and the third line defense, and inflammation is one of those things. I mean, we can think of inflammation, the chemicals of inflammation, being a lot like the bat signal that gets shined up in the sky to let Batman know that Gotham City's in trouble. So, you look up in the sky and you see, I haven't seen a modern Batman movie in a long time, but I used to watch the old show from the 60s with Adam West. Yeah, all of that. Those were great. So cheesy, but so much fun. But yeah, when they would turn on the bat signal, it would shine up in the sky, and then that was the signal that Batman needed to activate. And that's very much what chemicals of inflammation do. So things like histamines, and there's other classes of chemicals called cytokines that are basically going to jumpstart cells of immunity to get into action, and off we go. And the other thing that inflammation is going to do is it's basically going to bring in chemicals that are going to start the repair process. So inflammation will set the stage for the reparative processes to begin. So inflammation is good, but sometimes inflammation can get out of control, and when that happens, we have to do things to kind of reduce it, like ice the area. As a holistic healthcare provider, my advice to patients were, let an inflammatory process go for, the first 24 to 48 hours. And then if you have persistence of the inflammation, start gently applying ice just to help contain the inflammation, just to help it along. Help the body evacuate all those chemicals, because they can create a chemical traffic jam in the tissues. Now, there are 3 basic stages of inflammation. And we're going to look at each one of these, but we're going to talk about them right here, at least state what they are. The first stage is the inflammatory chemical release. So basically, these are the invitations to the party. And the second thing is that we're going to end up with vasodilation and increased vascular permeability. Now, if we have increased vasodilation and vascular permeability, basically what we're doing is we're making room for all these extra things to show up, because they're coming by way of the blood. That's the only transport system that we really have to transport these chemicals and cells of immunity around the body. So we have to, first of all, expand the vessels that are carrying the blood, that are bringing all of these things to us. And then the next thing is that we will be mobilizing all of these phagocytes. So these are the 3 basic stages of inflammation. So let's have a look at each one of these. Now, there are a number of chemicals that can be released, and there are classifications of chemicals. So this is truly a rabbit hole that we could go down, and there is so much to this part of the lecture. But I'm going to try to keep it very surfacey, so that we just get a general understanding of what's happening here. So the first thing that we're going to have happen in the inflammatory process is the chemical release, because the chemicals are going to be the invitation to the party. So what sort of chemicals get released? Well, it's going to be things like histamine, for example. That's a big player here. And these chemicals are going to be released into the extracellular fluid, the fluid that is outside of the cells. So it's going to be things like the interstitial fluid. It's going to be the fluid that is in plasma. So that's where we're going to see these chemicals moving into and taking their greatest effect. Okay, so that's what we're looking for with these. Macrophages and epithelial cells of boundary tissues are going to bear these special pattern recognition receptors called toll-like receptors, and once these get activated, they too will release certain inflammatory chemicals, but they're going to release something called cytokines that also help further inflammation. Now, the word cyto means cell, and kine, K-I-N-E, think of kinesiology, which is movement. So this classification of chemicals called cytokines basically is going to cause cells of, in this case, immunity to mobilize and do something. So it's a very general classification, and so in the spirit of general classification, I'm giving you a very general definition of what these cytokines are going to entice cells of immunity to do, which is basically get up and do something. We got a problem. Now, we know histamine is a big player in the inflammatory response, but there are other chemicals that can be released by these cells of immunity as well. So for example, kinins, another mobilizing sort of a protein, prostaglandins, and oh, look, complement. We know all about that. The Big Mac attack is what we get when we activate complement. So all of these have the capacity to be engaged with inflammatory chemicals and be inflammatory chemicals themselves. All of these are going to cause vasodilation of local arterioles, which is basically going to allow greater blood flow to the area, which means more immune players, immune cells, and immune chemicals, and which are going to release more kinins and prostaglandins and complement, and cytokines, which means more blood coming to the area with more players. So it's like this is truly a positive feedback loop that we see happening here. All right. Now, all of these are going to make the capillaries leaky. So once the blood gets there, we want what's in the blood to get out. So we're going to make sure our capillaries are good and leaky. Anything that wants us to spill out as far as immune stuff can certainly do that through our very leaky capillaries. Many of these chemicals are going to attract leukocytes to the area, so that'll be things like other macrophages, which is a white blood cell, monocytes, macrophages, neutrophils, maybe even basophils and eosinophils. So like I said, invitations are starting to go out to everybody, and everybody's starting to show up. Now, some of these chemicals are going to have other inflammatory roles, such as triggering pain receptors. Prompting the release of more inflammatory chemicals, because that's something that's happening all the time once this process gets going. Okay, so that was our chemical release. The next thing that's going to happen is this vasodilation and vasopermeability. So what's happening here is that we are bringing in extra blood to the area, and we want what's in the blood to slip out. Now, vasodilation is what is going to create something called hyperemia, which is basically extra blood flow to the area, or congestion with extra blood flow to the area. And when blood shows up, blood's going to bring with it its red color. So it's going to turn the area red. It also is going to bring with it its heat, which is going to turn the area hot. hot to the touch. So when you look at an injury site that is red and hot, then you're looking at that vasodilation and vascular permeability actually happening. All right. Now, with increased capillary permeability, we're going to create what's known as an exudate, which basically means fluid containing clotting factors and antibodies is going to leak out into the tissues. Now, we do want those clotting factors and antibodies in there because it is typically outside of the capillaries and in the tissues where we need those things to be, because that's where the damage is, right? Now, when we have all this extra blood flow to the area, that's going to lead not only to redness and heat, but it's going to cause localized swelling, which we can call edema. Now, swelling is our indication that we've got inflammation going on. I mean, redness and heat do too, but definitely swelling lets you know that you've got a chemical traffic jam starting to take place there, because there's all that extra fluid in the tissues, and that it might start slowing down the flow of fluids that can replenish the things that are needed, like maybe clotting factors or nutrients or whatever might be needed to do the reparative processes. The other thing that is happening there, and which is a bad thing, which is one of the reasons why we want to contain inflammation. Now, another thing that's happening with all this fluid accumulation out in the tissues is that it's putting pressure on the tissues. And this extra pressure can put pressure on the free nerve endings and create pain. And pain is often perceived as something of a good thing because it lets us know that we've got a situation that needs to be tended to. So that's why swelling in the area can lead to that discomfort or pain, is because all that extra fluid is just pressing on the nerve end, pressing on the free nerve endings, which are pain receptors, one of the pain receptors. Now, pain can certainly result from a buildup of fluid and the pressure that that's going to create, but pain can also be a result of toxins that might be being released by the pathogen that has invaded our tissues, because many of the chemicals that pathogens create are going to be irritating and noxious to our own tissues. Now, although I kind of give inflammation a little bit of a bad rap, it does have some advantages. So, for example, the surge of fluid in the tissues can actually act as a hose to kind of cleanse the area of any foreign material or any sort of garbage that might be in the area. So just sweep that stuff out, and it can be taken up by the lymphatic vessels and the lymph nodes and dealt with there. The other thing that this extra blood flow to the area is going to do is deliver clotting proteins and complement to the area, as well as clotting factors and fibrin. If we should, fibrin is going to be important, particularly if we're bleeding. So yeah. By bringing extra blood to the area, it's going to be bringing all the things that blood contains, like antibodies or white blood cells or the fibrin to stop the bleeding, or platelets to also help stop the bleeding. So yeah, because the blood is a major transport mechanism in the body. So of course, when we bring more of that to the area, we're going to be bringing more of what it's carrying to the area. And that's actually a good thing, at least in the short term. With all of this blood coming to an area, thanks to the vasodilation, we need to kind of pause and talk about the 4 cardinal signs of acute inflammation. And we've talked about some of these already. Actually, we talked about all of these. We talked about redness, heat, swelling, and pain. And sometimes there's a 5th sign known as impairment of function. And I believe that gets taught more and more. So we are going to include it here. So why do we have redness? Why do we have heat? Why do we have swelling? And why do we have pain? Well, and why do we even have impairment? Well, largely it's because of the extra blood flow to the area. So the extra blood flow to the area is going to create that redness. The heat comes as a result of extra blood being routed to the site of injury. We're going to end up with fluid spilling out of the bloodstream and into the interstitial space where the damaged tissue is, and that's going to lead to swelling. As that tissue tends to build in pressure, thanks to all that extra fluid, we're going to get pain. And if our swelling and pain are enough, we're not going to want to move that area, and that's going to lead to our 5th sign, which is the impairment of function. So those are your 4/5 signs of acute inflammation. And now we're at fever, which is our last innate defense that we're going to be talking about here. And fever is that unusually high, higher than normal body temperature that which is a response to some sort of an infectious process. And what happens here, the reason why we create a fever is that we have white blood cells in macrophages, which is a white blood cell, releasing these proteins known as pyrogens. And pyrogens are going to make their way up to the hypothalamus. And you remember the hypothalamus is the organ up in the brain, the region up in the brain that is going to be responsible for setting the body's thermostat or setting the body temperature. Now, pyrogens are going to allow that temperature to creep up. It's going to allow the body to become hotter, and there are some advantages to that. One of the benefits to a slight fever or a moderate fever, not a super high fever, but something within modest range, is that it can increase the metabolic rate of our cells. And when our cells metabolic rate is allowed to increase and the cell is functioning faster than usual, and that means that its rate of repair is faster, and maybe its rate of production of chemicals that are going to help fight pathogens might even be a little bit greater as well. So when a cell is being stimulated to do what it normally does to defend the body, and is allowed to do that faster and stronger as a result of pyrogens, well, then we're going to get a better response out of the immune system. The second thing that fever does is that it basically causes certain organs in the body to withhold nutrients that the microorganisms need to reproduce. So specifically, the liver and the spleen are going to be triggered to withhold iron and zinc. Iron and zinc. And these are two nutrients, I'm going to call them nutrients, that many microorganisms need in order to replicate. So basically it acts like a prophylactic for that microbe. So they don't have what they need to make more of themselves, and so it gives our body a little bit of an advantage to discover them and then kill these microorganisms before they can replicate.